So the medical problem I wanted to address in my thesis is cardiovascular disease. This is an umbrella term that characterizes diseases of the heart and blood vessels. These include diseases like myocardial infarction, also known as heart attacks, congenital heart defects, which are malformations of the heart, and also arrhythmias, which are irregular heartbeats. Diseases of the blood vessels include stroke, pulmonary emboli, and deep vein thromboses. You've probably known someone with one of these diseases, and that is because it is the leading cause of death in the world. And more specifically in the United States, it's the leading cause of death amongst all other causes for all races and biological sexes. So this is a problem that affects our longevity as a species. One of the reasons why it's such a big issue is because we don't have many good ways to study human cardiovascular disease. For one, it's unethical and illegal to study on live human hearts, and information we can get from humans requires functional readouts. We don't really know what's going on at a cellular and tissue level with high magnification. So as an alternative, for many years, we've used animals to study cardiovascular disease. These animals include zebrafish and all the way up to large animals like sheep and even non-human primates. Animals are good because you can study the progression of cardiovascular disease over time, but they are animals. They are inherently different than us. For example, cardiovascular disease isn't even a cause of death naturally for many of these animals, so we must induce cardiovascular disease in them. And this manifests in the fact that over 90% of drugs tested in animals go on to fail in clinical trials. Furthermore, the larger the animal, the more expensive it is to maintain, and the longer duration of time it gets to a mature point where we can actually study cardiovascular disease where it models human cardiovascular disease. So, as another alternative, researchers have used what are called new approach methodologies. These are human-based models that utilize human cells and human tissues to study the cellular and tissue function of human hearts. One of these models relies on human pluripotent stem cells. Human pluripotent stem cells can make any cell in the body. They were first successfully cultured in 1998 when a group successfully harvested the inner cell mass of a developing human embryo and cultured it in a dish, allowing them to culture these cells in a dish. And then a follow-up study utilized proteins that were highly expressed in these human embryonic stem cells and encoded them in a virus and infected human skin cells, and these transformed these cells into human pluripotent stem cells. This work went on to win the Nobel Prize. And the unique thing about this type of human pluripotent stem cells, iPSCs, is that they allow for the study of patient-specific cell types. So if I take your cells, I can turn it into a human pluripotent stem cell and then make any cell that I want to, basically, found in you, basically. And so if there is a genetic disease that particularly affects an organ of interest in you, we can make that using your cells. Another cool feature of human pluripotent stem cells is they can be cultured indefinitely. So once you have one of these, as long as you feed them the right conditions, you can culture them forever, basically providing a never-ending source of human cellular material. And since human pluripotent stem cells were heavily utilized, many groups around the world have developed protocols to make specific cell types, basically all of them in our body, including germ cells, ectodermal derivatives such as neurons, mesodermal derivatives such as cardiomyocytes, like muscle cells of the heart, and endodermal derivatives. And then starting in the in the 2010s, groups started to make what are called organoids. These are 3D structures grown from stem cells that self-organize through cell sorting and spatially restricted lineage commitment in a manner similar to in vivo organogenesis. So what that looks like in practice, you take pluripotent stem cells, you utilize differentiation and bioengineering techniques, and you generate miniature organs, basically. These are termed organoids, and they are structurally and functionally similar to organs found within us. Because they are derived from pluripotent stem cells, many groups extrapolate findings from the creation of these organoids to organogenesis, or the creation of organs. And because these organoids structurally and functionally mimic organs found within us, many groups use them for disease modeling. The next progression of this technology is called assembloids. These started to appear in the late 2010s to this current day. And these are a self-organizing cellular system resulting from the combination of a type of organoid with another type of organoid, or with a different specialized cell type that result in integration. So what this looks like in practice, if you differentiate an organoid from pluripotent stem cells, you take those same pluripotent stem cells and differentiate another cell type or another tissue type that is generally missing from the tissue, and then you combine them to form an assembloid. These have higher physiological complexity because they generally feature a cell type that should be there that previously wasn't. And the benefit of adding a missing cell type or a missing tissue type is you can actually study cell-cell and cell-tissue interactions based on now the inclusion of that cell type and how it affects the development and homeostasis of the tissue. And so I sought to create an assembloid. Back in 2020, I joined the Geary lab, and they were pioneers of one of the first human heart organoid differentiation protocols. To make them, they dissociated pluripotent stem cells, aggregated them into what are called embryo bodies, and subjected them to different chemical inhibitors and growth factors to manifest a cardiac phenotype. Here's a beating heart organoid. As you can see over time, there's stronger expression of this protein that's expressed in cardiomyocytes called cardiac troponin. And as you can see with development, it gets stronger and stronger in expression. They characterized the cellular content of these organoids, and they found that not only did they feature cardiomyocytes, but other cell types found within the heart organoid, including endocardial cells, endothelial cells, epicardial cells, and fibroblasts. Not only did they have these, but they also organized in a way that mimicked how our hearts are organized. They had epicardial cells on the outside of the organoids, they had endothelial cells perfusing through the tissue, and they had chambers lined with endocardial cells. This was one of the most advanced models of the heart at the time. However, it's missing a cell type, and these are immune cells. Immune cells are found all within our body, and they are derived from a different developmental process called hematopoiesis. And so it derives red blood cells and all the different white blood cells or immune cells that our body has. One of the cells that we wanted to model first were macrophages, and primarily because macrophages are in every tissue in our body. Macrophages exist on a spectrum of function, and these are specialized immune cells within our innate immune system. On one side of the spectrum, they participate in leukocyte recruitment, activation of vascular endothelial cells, leukocyte activation, and also communication with our adaptive immune system to leverage a remembered immune response. Macrophages that participate in these functions are typically termed recruited, pro-inflammatory, and they're derived from the bone marrow, and they evacuate tissue generally post-inflammation, and they are positive and express this protein called CC chemokine receptor 2. On the other side of the spectrum, they also can produce anti-inflammatory factors, dampening the immune response. They clear dead cells. They inhibit leukocyte recruitment, and they also participate in tissue repair and extracellular matrix remodeling. Macrophages that participate in these functions are generally termed tissue resident, anti-inflammatory, or pro-resolving, derived from the yolk sac or fetal liver, and they persist in our tissues throughout our entire lives. And generally, they are CCR2 negative. Keep note of that. We wanted to model tissue-resident macrophages due to the fact that our organoids, since they are derived from pluripotent stem cells, which mimic embryonic stem cells, mimic the developing human heart. And so, if we added these tissue-resident macrophages, which seed which seed tissue during development, then we would faithfully model these tissue-resident macrophages. So, what do they do in the heart? So, all we know about what they do in the heart is from animal models. So, in mice specifically, they participate in coronary patterning. If you look at these sections here, you have a control mouse and then a macrophage-opposed mouse, and you can see these vessels here patterned in a different way. And you can also see this in the perfusion scan here. They also play a role in valvular development and remodeling. In red and green, you can see macrophages associating with the aortic valve through development of this mouse and the mitral valve. They also play a role in lymphatic channel development. Here, in knockout macrophage mice, you can see that these lymphatic channels are truncated, and they're not as plentiful as the control mouse. They also participate in homeostatic functions, which are the body's process of maintaining itself. They play a role in eliminating what are called cardiac exophores, which are basically extracellular vesicles secreted by cardiomyocytes containing byproducts it no longer needs, and the macrophages help eliminate those. They also limit fibrosis and promote angiogenesis, or the formation of new blood vessels after injury. And they also facilitate electrical conductance. Here you can see the AV bundle, which is a structure in the heart's conductance system, and you can see in green lots of macrophages associating in this area. They quantified this, and they actually found that macrophages seem to preferentially localize to this area versus a more muscular part of the heart, such as the left ventricle free wall. So this implicated their role in electrical conductance. And so we know all this about tissue-resident macrophages in the heart, but again, these are mice. But we don't actually know what they do in humans. So we wanted to explore what the role of human cardiac tissue-resident macrophages were in the heart. However, we know they are there. And so this is single-cell RNA sequencing data tracking cell populations over time in the developing human heart. As you can see here, in 5 to 7 post-conception weeks, macrophages make up 2.5% of all cells, and they grow in population over time. And so we thought, if they are there, they must be doing something. And so in order to study that, we needed to actually create a model to see what they do. And so our first aim for my thesis was to engineer a human heart macrophage assembly. Going back to our hematopoiesis chart, we can see that macrophages are down here, and they are derived from a precursor called monocytes. Monocytes are a circulatory form of macrophages, and we hypothesized if we add these to the heart organoids, they would bind, differentiate, and integrate and migrate and take residence in the tissue. And so that is what we hypothesized. And so what we did is we differentiated monocytes from human pluripotent stem cells using different chemical inhibitors, growth factors, and cytokines to generate monocytes. In this column here, you can see an H&E, or hematoxylin and eosin stain of monocytes, and you can see they're about 10 microns in size, circular, have very large nuclei, and that top one has a nice distinct kidney shape, very nucleus characteristic of monocytes. They also express CD14 and CD68 monocyte markers. And we also did flow cytometry, a common technique for analyzing immune cells. And you can see that they express 94% of them, CD14, CD45, CD163. And then we looked at that CCR2 marker, which tissue-resident macrophages should not express, and about 99% of our monocytes do not express that. So we thought if we add them, they would remain that way and take on a tissue-resident macrophage phenotype. Now that we created our monocytes, we needed to figure out when and where exactly we wanted to add them to the organoid. So to do this, we used developmental phenomena that occurred during mouse and human heart development. As you can see here, macrophages begin to seed the mouse heart between embryonic day 10 and embryonic day 12. And in the human heart, we don't know when they're first evident, but we know 2.5% of all the cells in the human heart by day 35 are macrophages. Looking at the events, we noticed that these two events flanked the presence of macrophages in mouse and human heart development. These are the initial heartbeats and then coronary artery formation, which is the blood vessel that feeds blood to the heart. And so when we looked at human heart organoid development and looked for analogous events, we found that these occur at day 6 and day 12 of human heart organoid development. We also did our, bulk RNA sequencing to look for genes associated with the following process. We found upregulated gene expression in macrophage chemotaxis, monocyte differentiation, and leukocyte adhesion in this day 7 to day 9 range, indicating that these organoids are essentially saying, hey, we're ready for monocytes. And so when we did immunofluorescence staining for one of these markers, VCAM1, on an organoid within this time frame, we saw that there was co-localization between this VCAM1 and TNN and T2, which are cardiomyocytes. So this supported that if we added monocytes, it would actually bind to the heart organoid in this time frame. We considered one more thing. There's a day 7 cheer exposure, which manipulates the Wnt pathway that helps promote epicardial tissue expansion, and we did not want to interfere with that. And so we decided to delay it by 1 day, and we decided to start adding monocytes at day 8 of organoid differentiation. So what this looks like in totality, we take our pluripotent stem cells, we differentiate some to become heart organoids, and differentiate others to become monocytes. Once monocytes are produced, we use magnetic antibody cell sorting to select for cells that express CD14, which monocytes express CD14. We then pipette resuspend these monocytes with the organoids at day 8, day 12, and day 16. And by day 20, we have what we call human heart macrophage assembloids. The next thing we wanted to do was characterize these assembloids. We tried different monocyte addition amounts per addition to see what would be the best method of integrating the monocytes and how many monocytes it would take to integrate the organoids to a physiologic level of tissue-resident macrophages. For one, we wanted to see where they localize. And so here we did immunofluorescence staining for our organoids, and the macrophages are stained by green. It's called CD45 is the protein that's staining for. And as you can see, there's green signal on the outside and inside of the organoids. We created a ratio of CD45 positive signal on the inside of the organoids versus the outside, with a ratio greater than 1 indicating more CD45 signal on the inside of the organoid. And we found no matter how early we started adding the monocytes were binding and differentiating and getting inside of the organoid, no matter how early we added them. So this indicated that the monocytes are binding and integrating inside the organoid. We then wanted to quantify the macrophage population. To do this, we did flow cytometry. We dissociated the assembloids into single cells and then stained them for a protein called CD45. As you can see, with greater additions and day, by day 20, we were able to get a physiologically relevant amount of tissue-resident macrophages inside of our assembloids using 20,000 monocytes per addition or 30,000 monocytes per addition. We decided to go with the 20,000 monocytes per addition going forward because, as you can see in the 30,000, it yielded some assembloids with too many macrophages in the system. We repeated this across 3 different pluripotent stem cell lines, including 2 embryonic stem cell lines and 1 induced pluripotent stem cell line, and yielded similar levels of integration. The next thing we wanted to do was prove that they persist in the assembloid for long periods of time. If they are truly tissue-resident macrophages, they should. And so what we did is we did immunofluorescence microscopy, culturing these assembloids for 60 days with no further monocyte additions. As you can see in the immunofluorescence staining, staining monocytes or macrophages with CD45 and CD163, we can see at day 60, we have this green and cyan signal still through day 60. And when we took a high magnification images of one of these assembloids at day 60, you can see a nice macrophage embedded in the myocardium of the assembloid. We then again used flow cytometry to quantify the CD45 cell population over time, and we noticed that it stayed within the physiologic relevant population of macrophages through day 60, indicating that they bind and persist in the assembloid through time. The next thing we wanted to do was single-cell RNA sequencing to actually validate that these are, in fact, tissue-resident macrophages in identity. So single-cell RNA sequencing was done by first dissociating the assembloids into single cells, and then we did transcriptional analyses. Here's a UMAP plot, and each dot on this plot represents the transcriptomic profile of one cell. When these dots are in close proximity to one another, it indicates that they are more similar to one another, and this allows us to cluster them and classify them. You can see tissue-resident macrophages are down here, none in the heart organoid. That's good. And then we did again, transcriptomic analysis looking for gene expression of these relevant genes with a dark blue color indicating high expression. And you can see that this macrophage cluster stains all these different relevant cardiac tissue-resident macrophage markers. Notice again, we did a CCR2 expression analysis, and it's completely grayed out, indicating no CCR2 expression. So this data confirmed that these are tissue-resident macrophages in identity. We then compared this data set to one of an embryonic human heart. And so on the left is that data set, and you can see with our cluster identities, they match and map onto that embryonic data set very well. Atrial, ventricular, endothelial, endothelial. They also have an immune cell cluster, but they didn't do a rigorous analysis to see what immune cells are actually in their system. But it mapped really well with our tissue-resident macrophage cluster. And so we did a differential gene expression analysis and looked at cells specifically that express CSF1R, which tissue-resident macrophages express in this immune cell cluster of human embryonic hearts and then our heart assembloids. And you can see, given the Pearson correlation coefficient, they had a really strong coefficient of 0.8, indicating that our macrophages are very, very similar to their tissue-resident macrophages in the heart. And so, at the end of this aim, we engineered a human heart macrophage assembloid. We proved that when we add these pluripotent stem cell-derived monocytes, they bind, migrate, and populate the organoids to form an assembloid. Incremental monocyte titrations achieved a physiologically relevant tissue-resident macrophage population in the assembloids. These macrophages persisted for long durations, and these tissue-resident macrophages resembled human embryonic cardiac tissue-resident macrophages. Now that we created our model, let's do something cool with them. Let's figure out what the tissue-resident macrophages are doing. So our next aim was to molecularly and functionally characterize the heart macrophage assembloid system. Our hypothesis was that the tissue-resident macrophages contribute to the development and homeostasis of the heart macrophage assembloid, resembling the functions of embryonic cardiac tissue-resident macrophages. The first function we wanted to prove is efferocytosis. Efferocytosis is a fancy word for immune cell gobbling up dead cells or cell debris within the tissue it resides in. To do this, we did a flow cytometry assay where we gated for live tissue-resident macrophages and looked for the staining of apotracker green, which is a dye that stains phosphatidylserine, which is exposed on the outer membrane of cells that are undergoing apoptosis or regulated cell death. So we gated for these types of macrophages that contain this material, because if they contain the material, that means they engulf them. And we tested and we quantified the amount of these apotracker positive macrophages in assembloids, organoids, and also the assembloids with an efferocytosis inhibitor. And we noticed when we added the efferocytosis inhibitor, we saw a reduction in the amount of macrophages that contain this apotracker green dye, indicating we were in fact inhibiting efferocytosis and showing that the macrophages participate in efferocytosis, given the perseverance of the apotracker green positive macrophages in the assembloids. The next thing we wanted to look at was the potential communication role of macrophages. Macrophages are immune cells, and they're constantly communicating with their environment to understand where they need to localize and where their function is needed. One of the ways in which they do that are they secrete little packages called extracellular vesicles. These contain proteins, RNAs, that allow them to communicate with other cells. The beauty of our system is that we culture the assembloids in media. And when we culture the tissue in the media for long durations, it actually secretes their packages into the media, and then that allows us to collect those packages, or extracellular vesicles. So we collected them, digested them, and did a proteomic analysis using liquid chromatography, mass spectrometry, and analyzed the peptides found within the media, and compared that to the extracellular vesicles found in the heart organoids. We found in this analysis that there were 150 unique proteins in the heart macrophage assembloids versus those in the 55 in the heart organoids alone, And this indicated to us that the macrophages were contributing very uniquely to the communication that occurs within the tissue. And when we looked to see what processes these proteins were involved in, we found that they were related to cell matrix adhesion, regulation of macroautophagy, and cytoskeleton organization. So this showed that macrophages likely play a unique communication role in the tissue. One of these cytoskeleton proteins is collagen. So the cytoskeleton is the scaffold that our cells reside upon that gives them and our tissue structure. And we found that in our transcriptomic analyses that comparing human heart macrophage assembloids to heart organoids, that the collagen fibril organization was altered, indicated by our transcriptomic data. To validate this, we did immunofluorescence microscopy imaging for staining of collagen 1, and you can see by the images above that the distribution of collagen is different between that of an assembloid, which has macrophages, versus organoids that don't have macrophages. We quantified the images and found that the macrophage assembloids had more collagen 1 overall, and we also used a plugin that measures entropy of signal. And so when we found that the macrophage assembloids had a lower collagen 1 entropy, meaning it's more organized, throughout the tissue, whereas in the heart organoids, it was more homogeneous, localized in certain pockets of the tissue. We also did real-time quantitative qPCR to validate this data, and this also found that the heart macrophage assembloids express more collagen 1. So this data indicated that tissue-resident macrophages play a role in the organization and expression of collagen 1. We also wanted to look at its impact on ventricular myogenesis. There was a paper that came out last year. It's an engineered heart tissue paper, which incorporated macrophages into their little engineered heart tissues. And they found when they added the macrophages, it actually pushed the distribution of cardiomyocytes from more of an atrial phenotype, which are the top chambers of the heart, to more of a ventricular phenotype, which are the bottom chambers of the heart. And we wanted to validate if that happened in our system. And so what we did is we looked back at our single-cell RNA sequencing data and looked at the distribution of these different cell clusters. And we found that there was an increase in the amount of ventricular cardiomyocytes in our heart macrophage assembloids versus that in the organoids. We also did gene set enrichment analysis and found that myogenesis genes were more upregulated in our heart macrophage assembloids versus those in the organoid. To validate this data, we then did immunofluorescence microscopy, staining for MIL3, myosin light chain 3, which is predominantly expressed in ventricular cardiomyocytes. And as you can see by the images, its distribution is much more throughout the tissue in heart macrophage assembloids versus the heart organoids. So this indicated tissue-resident macrophages play a role in ventricular myogenesis in our system as well. The next thing we wanted to do was look at contractility. In the same engineered heart tissue paper that I referenced in the last slide, they also found incorporation of macrophages enhanced the contractile force of their tissues. And so using a Fiji plugin called Muscle Motion, which tracks the motion of tissue as it contracts, feeding it live cell video, we can see that the heart macrophage assembloids beat much stronger than these heart organoids, and so much so it manifested with significantly higher contraction amplitude. We also did transmission electron microscopy, looking at the sarcomeres, which are the contractile units of the cardiomyocytes, and we found that they were much shorter in the heart macrophage assembloids than the organoids, indicating that our assembloid cardiomyocytes were in a more contracted state overall. So this showed us that the tissue-resident macrophages impact the contractility of the human heart macrophage assembloids. The next thing we wanted to do was look at the tissue-resident macrophage's role on electrophysiology. So macrophages, when they bind to cardiomyocytes, they form what are called gap junctions, which are pores that facilitate the transmission of signals like ions through them. And so in green, we have macrophages, and in white, we have one of these gap junction proteins called connexin 43. And you can see that it's co-localized over the cardiomyocytes here in this panel, and it's indicated by yellow arrows, these gap junctions. We also have an example of macrophages that didn't form these gap junctions, which is indicated by this white arrow here. And so we quantified the distribution of these macrophages, whether they were connected or they weren't connected to the myocardium. And we found the majority of them were connected to the myocardial components of our heart macrophage assembloids, implicating their role and participating in electrophysiology in our system. We also did real-time quantitative qPCR for gene expression of amphoregulatin, which is a protein that's expressed as a result of gap junction formation between macrophages and cardiomyocytes. And we saw that that was significant in our system. Now that we proved that gap junctions form within our system, we wanted to prove that they were actually functional. So the next thing we did is live cell imaging. We used different ion dyes, one called Fluophore, which shows calcium fluxes, and then we have one called Fluovolt, which is a membrane voltage dye, which shows changes in action potential. membrane voltage. So differences in membrane voltage result in a flash. And so here you can see macrophages standing in M cherry overlying a region of the myocardial component of our heart macrophage assembloids. And as you can see, these macrophages are synced with one another with their neighboring adjacent cardiomyocytes. So with each flash, that signal is being propagated through these red areas, which are our macrophages, showing that our macrophages actually accept the signal from its neighbors. We then wanted to see how the macrophages handled the ions in the system. And we found that tissue-resident macrophages actually had prolonged action potential durations as compared to cardiomyocytes. So this shows that tissue-resident macrophages handle the signal much differently than cardiomyocytes, implicating them for a potential role in facilitating electrophysiology in our system. And so we characterized the function of these tissue-resident macrophages in our system. We showed that they partook in efferocytosis, altered the proteome of the extracellular vesicle content, increased organization and expression of collagen, changed cell populations, impacted ventricular myogenesis, and facilitated electrophysiology and contractility in our system. And so with that, we wanted to do one more thing with it. So our third aim is to define the role of tissue-resident macrophages in NLRP3-driven atrial arrhythmias using heart macrophage assembloids. And again, if you don't know what that means, I'll unpack that. So for aim 3, we wanted to figure out, or I wanted to use it for a clinical purpose to study something clinical that someone could potentially benefit from in the near future down the road. So I was looking for inspiration of what to use this model for. I joined the lab during COVID, so a lot of weird things were going on during COVID. And I am a kind of a sports fan, so there were some pretty high-profile incidences that occurred during my time starting out in the lab. One of which was during the Euro 2020 championships when Denmark's main man collapsed all of a sudden during a game. It was actually being played in 2021 because COVID kind of shut everything down in 2020. And so his heart stopped completely, and he was able to be brought back to life on the field and eventually returned to playing soccer. But that was a scary moment. Shortly after that, one of the best strikers in the world, Sergio Aguero, went up for an innocuous header in a game for one of the best clubs in the world, and then came down and went into atrial fibrillation and was forced to retire from soccer altogether. A colleague of mine also passed away shortly after this, and his cause of death was unknown. When someone can't find a cause of death, it is usually attributable to a fatal cardiac arrhythmia. And so this made me interested in the potential relationship between inherently pro-inflammatory events that causes systemic inflammation, like COVID-19, and heart function. And so when I looked at the literature, I found that atrial fibrillation was one of these arrhythmias where inflammation could play a role. Atrial fibrillation occurs when the top chambers, atria, misfire and don't communicate with the lower chambers really well, called the ventricles. This manifests in an irregular heartbeat. It affects over 50 million people worldwide, and if you don't get it treated, it likely will result in death, stroke, or heart failure. The treatments include calcium channel blockers, beta blockers, and cardiac ablation, but these treatments aren't very good for long-term treatment. If you get atrial fibrillation, it is likely to come back, and that is called recurrent atrial fibrillation. And generally, cardiac ablation is the only procedure that can treat this. However, it is only 50% effective over 5 years, and so it is likely to come back. Furthermore, there's been limited pharmacological advancement to treat atrial fibrillation over the past 40 years. So to this day, atrial fibrillation remains a problem, and there aren't many good therapies to treat it. You're likely to get atrial fibrillation if you have one of the following risk factors. Old age is the common one, hypertension, diabetes, the usual suspects. But in my literature search, I found inflammatory events actually increase the likelihood of you developing atrial fibrillation. One of these diseases called sepsis. When you are hospitalized with sepsis, you actually have a 25% chance of developing new onset atrial fibrillation. So this implicated a relationship between inflammation and cardiac arrhythmias such as atrial fibrillation. And so in 2018, a group found that the link between inflammation and these atrial fibrillation is something called the NLRP3 inflammasome. The NLRP3 inflammasome is a large intracellular protein complex that forms as a result of pro-inflammatory stimuli binding to the outside of atrial cardiomyocytes. These pro-inflammatory stimuli include interleukin-1 beta, TNF-alpha, LPS. And then this causes a transcription cascade, upregulating expression of NLRP3, and then pro-inflammatory cytokine precursors, such as interleukin-18 and interleukin-1 beta. Once the NLRP3 inflammasome activates, it oligomerizes, so it binds with these different protein components, ASC and caspase. And once it's activated, the caspase then cleaves these pro-inflammatory cytokine precursors into their active forms, which secretes them outside of the cell to propagate the inflammatory response and also causes cell death. In the same study, they looked at patients with chronic atrial fibrillation versus age-matched controls, and they did a Western blot and found that these components of the NLRP3 inflammasome were significantly more expressed as compared to the controls, implicating the inflammasome and atrial fibrillation disease. However, these were postmortem samples. They didn't prove that the NLRP3 inflammasome was causal to this phenotype, and so they were in no position to show that. But with our system, we can show that. Our human heart macrophage assembloid models live human hearts. It contains spatially organized atrial and ventricular compartments. It has measurable electrophysiology and features tissue-resident macrophages. And so with our system, we figured that activation of the NLRP3 inflammasome in the atrial cardiomyocytes of our assembloids induces an atrial fibrillation-like phenotype. So for this experiment, what we did is we subjected our macrophage assembloids to maturation conditions to get our assembloids to the most advanced physiologic age as we possibly could, because atrial fibrillation affects predominantly those who are of old age. Once we got them to that stage, we then exposed them to different pro-inflammatory factors that cause NLRP3 inflammasome activation, such as LPS, interferon gamma, and interleukin-1 beta. And we added them to the media for 30 days. We tried 3 different concentrations of each factor to add, with low being the pathophysiologic concentration of these factors in our blood in an infection or something like sepsis. And then medium dose being 2 times that, and the high dose being 20 times the low dose. And what we found using, again, that FluoVolt live cell microscopy is that our control assembloids in the atrial compartments be nice, really regular. But once we started adding these pro-inflammatory factors, the rate increased substantially. We also noted ectopic events where there was skipped signaling. And then with higher doses, we saw more and more of these irregular phenomena. We quantified this and we found that the beat rate overall was significantly higher no matter how low the pro-inflammatory factor concentrations were. And the incidence of spontaneous irregular rhythms and waveforms were much higher in the medium, low, and high doses versus the control. One of these irregular waveforms is called early after depolarization. It's a hallmark of atrial fibrillation disease pathology. And as you can see, when there's a normal depolarization and on the repolarization, there's another depolarizing event. That is an early after depolarization or an EAD. Again, we quantified this and we found in the medium dose, it was significantly higher as compared to the control. So we were manifesting an atrial fibrillation-like phenotype in our assembloids. The next thing we wanted to do was show that we were actually activating the inflammasome, the thing we set out to activate. To do this, we needed to show both steps, that priming step, which triggers the transcription cascade, and then activation. To show that priming occurred, that first step, we looked at and did immunofluorescence staining of NLRP3, which is a component of the inflammasome, but it should increase in expression with priming. And as you can see in atrial cardiomyocytes stained with NR2F2 and F-actin, we can see that there is more NLRP3 staining in the atrial cardiomyocytes versus the control. We then wanted to quantify ASC specs. ASC is one of the components of the inflammasome, and when it activates the inflammasome, it actually oligomerizes and forms these gigantic things called ASC specs, which are 1 micron size signals within our atrial cardiomyocytes, stained now this time with Mil7. And we quantified these specs in the atrial cardiomyocytes, and we found that there was a higher incidence of these specs in atrial cardiomyocytes amongst the medium dose condition versus the control. So this proved that we were activating the inflammasome. The next thing we needed to do to actually prove that it is causal to the phenotype, though, is we need to inhibit it in some way. So we used one of these inhibitors called MCC950 to prevent the formation of the inflammasome. So again, we repeated those same experiments, this time with the inhibitor, and we actually found the beat rate didn't change very much. But we did find the incidences of spontaneous irregular rhythms was significantly decreased, and one of these irregular waveforms, the EADs, was also significantly decreased. So this showed that the NLRP3 inflammasome, while it didn't affect rate at all, it did improve the incidence of irregular rhythms in our system, indicating that inflammasome inhibition is partially causal to the atrial fibrillation phenotype in our system. Again, looking just to confirm, we inhibited the inflammasome, we did immunofluorescence microscopy, and we saw a significant reduction in these ASC specs in the inhibited condition. Now we wanted to investigate the role of tissue-resident macrophages. We added them at the beginning, went all through that trouble to add them, right? So what are they going to do in the system? A paper came out last year which created an atrial fibrillation model by adding interleukin-1 beta to the mouse, or injecting it to the mouse every day for 15 days, and this made the mice more susceptible to developing atrial fibrillation in mice. So very similar to our experimental setup. And they found using different transgenic models that if they knocked out the receptor that binds interleukin-1 beta, basically rendering the resident macrophages non-functional in the mice, that it actually rescued the atrial fibrillation phenotype. As you can see here, they have a high number of atrial fibrillation episodes in the wild type with interleukin-1 beta condition, and then in the knockout transgenic mice model of tissue-resident macrophages, a complete reduction in the inducibility and number of episodes. So we hypothesized in our system that the tissue-resident macrophages under chronic inflammation would polarize to a pro-inflammatory state and increase the atrial excitability of our assembloids. And so for this, we used organoids, which don't have macrophages, as a control, and expose them either to control or medium dose of the pro-inflammatory factors. As you can see by the traces, the rate is much faster and there is much more ectopic activity. But when we quantified this, the beat rate was actually significantly higher as compared to the heart organoids, which don't have macrophages, versus the assembloids that do. So that was significant, but the incidence of spontaneous irregular rhythms wasn't that significantly different, slightly increased, but not significant. So this showed us that that macrophages, when included in the system, actually accentuate the atrial excitability of our tissues, causing this faster rate. To see how the tissue-resident macrophages might be inducing this effect, we did real-time quantitative PCR to look for gene expression of genes related to the following processes: NLRP3 inflammasome activation, secreted pro-inflammatory cytokines, and anti-inflammatory cytokines. And we found in the blue condition, which is our medium dose, the pro-inflammatory condition versus the control, looking specifically at the tissue-resident macrophages, we see an increase in genes related to inflammasome activation and pro-inflammatory cytokines, but a decrease in anti-inflammatory cytokines indicated over here. So this indicated we were actually polarizing and pushing the tissue-resident macrophages to become more pro-inflammatory in phenotype, and this might be how it's achieving that increase in excitability of the atrial tissue. And then now, just to validate that, we did immunofluorescence microscopy, again looking for ASC specs, and we found that there are more ASC specs in our medium dose within the tissue-resident macrophages themselves in our assembloids versus the inhibited condition, showing that our macrophages had the NLRP3 inflammasome activated within them as well. And so, in summary, we defined the role of tissue-resident macrophages in this NLRP3-driven atrial arrhythmia using human heart macrophage assembloids. We created the first protocol for modeling inflammation-induced atrial fibrillation. We partially resolved the phenotype through NLRP3 inflammasome inhibition and polarized the macrophages to a more pro-inflammatory state under these conditions. And lastly, we showed that the tissue-resident macrophage presence increased the atrial excitability with this chronic inflammation. And so, in totality, we engineered an immunocompetent human heart macrophage assembloid to explore the impacts of tissue-resident macrophages on cardiogenesis and homeostasis. We determined that the macrophages in our human heart tissue contribute to efferocytosis, secrete extracellular vesicles with unique proteomes, contribute to extracellular matrix organization, ventricular myogenesis, synchronize with native electrophysiology, and enhance contractility. And we developed an NLRP3 inflammasome-mediated human atrial arrhythmia model. And within this model, we found that tissue-resident macrophage presence enhances the excitability of the atrial tissue. Color-coded in black, these are novel contributions and findings in science. In red, these are findings that validate previous new approach methodology studies. In blue, this validates findings from animal studies. And in purple, this validates findings from both these types of studies. So in the future, I hope two things. One, I hope someone either uses my system or someone else's system to understand what the extracellular vesicle contents are for the macrophage assembloids and what they do, so we can model and basically mimic, whether it be a cure or something, or I don't know, a therapy or something. You can model these signals and then add them to your system and see how they impact the overall tissue. So this could be a cool approach for therapeutic development. I also hope that someone explores maybe clinical trials for NLRP3 inflammasome inhibitors. As I showed, partial inhibition of the inflammasome results in partial rescue of the atrial fibrillation phenotype. So this could really benefit patients with sepsis, autoimmune disorders, myocarditis, pericarditis, things like that. And yeah, I guess this is more for the committee. These are the publications I did while I was here. I was productive, I promise. In bold, that's predominantly the work I presented to you today in bold. These are the pending patents I contributed to during my time here. These are all the presentations I did and plan on doing in the next few months. And this is some of the professional service I contributed to. I was a reviewer for a bit for Frontiers, and also did a fun laboratory instructor experience out in Holland, Michigan in 2024. That was a lot of fun for stem cell and organoid biology. I see a few of you here. Hey. And then these are some of the awards and honors I received while I was here. And then I'd just like to thank a bunch of people. So thank you, Aitor, for pushing me to be the best that I could possibly be. Without your mentorship, I definitely wouldn't be here right now. Thank you to all the members of the Aguirre Lab, old and new. I've been here for quite a while, so a lot of turnover. But shout out to the graduate students, technicians, postdocs, undergrads, and medical students who all contributed to the work that you saw today. I'd like to thank the DO PhD administrators. That's the program I'm part of. Thank you for taking a chance on me and accepting me and guiding me and mentoring me throughout this process. I'd like to thank all our collaborators or all the people I got to collaborate during my time here. Even if it didn't manifest in a grant or a publication, I still had a good time. So thank you. It was good learning from you guys. I'd like to thank my committee members, Dr. Gary, Dr. Spence, Dr. Basil, and Dr. Zinn. I hope you liked what you saw today. And then I'd also like to thank LabRassist Incorporated, a startup I started while I was here. And I'd like to thank Simon K., Maxwell, and Dr. Vilkins for contributing to that experience.